Hello and welcome to the DDW sponsored vodcast series, where we examine the people, the breakthroughs and the innovation driving the drug discovery and development sector. I'm Megan Thomas, Multimedia Editor at DDW. DDW is the multi-platform global voice for the drug discovery and development community. From research stage to clinical trials, the brand covers the latest innovation and expertise in the sector. Above all, DDW readers learn how to turn science into business. Today's episode is sponsored by the Jackson Laboratory, and our guest is Adriano Flora, who I will be interviewing on the topic of alternatives to non-human primates. First, a little bit about today's guest. Adriano Flora currently serves as Director of Business Development at the Jackson Laboratory. He obtained his PhD in Pharmacology and Toxicology from the University of Milan, after which he moved to the Baylor College of Medicine in Houston, Texas, to study the molecular mechanisms underlying cellular differentiation and tumorigenesis in the nervous system. In 2019, he joined the Jackson Laboratory and has since been involved in developing and commercializing translational platforms for drug developers. So thank you for joining me today. The first question I have for you is, what is a humanized mouse model? What are the key benefits of this, and what impact will they have on the future of drug development?
Okay, so first of all, thank you for having me today. And what is a humanized model? It's kind of a very large question. A humanized model, so to speak, is essentially a mouse model that express a human feature. It could be either a genetically humanized mouse model or a mouse model that has been humanized with human cells. In a specific case, and what we will be mostly talking about, these will be humanized models that have been humanized with human immune cells. What are the benefits for drug development? Well, we will discover those, I believe, during this chat. But just for a high-level answer, benefit is that you reproduce specific aspect of the immune system, the human immune system, in an in vivo model that therefore has an holistic view of what happens to the immune system when you challenge it with your drug. That's the main benefit. So you can predict some aspects of how patients will respond to your drug.
Excellent. Thank you for that answer. That sets us up really well for the rest of this conversation. The next question I have for you is, how accurately do they mimic human physiology and immune responses?
Oh, that's a loaded question in a sense, because the accurately part can be expanded as much as you want. The most important thing to remember, these are models. Like all models, they will mimic very well some aspects of what you're studying, and they will not mimic other aspects. Humanized models come in very many different flavors and forms, and we can get into more details later on. But I would say that for aspects such as cell functionality, depending again on the model, you will have very, very good relationship between the model and the human being. For example, T cells are very much functional in these models, just as they work in human beings. Other aspects that are more complex might be lacking. For example, antigen presentation is still not perfectly reproduced in this model. So depending on what your needs are, what the mechanism of action of your drug, this model might be pretty accurate or might not be accurate. And this is one of the theme, I hope, of this chat, which is you really need to know how your drug works before you choose the model, because these will really affect how the results will impact then your research.
Great, thank you so much. So this next question I have for you is actually quite a complex one, so I'm going to break it up into smaller parts rather than load them all on at the same time. So the first part is, can you elaborate on why the preclinical development of antibodies and most biologics suffers from the lack of predictive in vivo models?
Maybe I'd like to elaborate a little bit on that, if I may, and because this is a true statement. Until now, the in vivo models that have been used classically for small molecule drug development really did not translate well for antibody development or biologic development. And you can see in the fact that most of these models have been dropped. The main reason is because an antibody interacts with its target protein through a surface, not like a small molecule will bind to a pocket in a protein by interacting with very few amino acids with more or less affinity. And therefore, these bindings are generally less specific than what an antibody will give you. An antibody, as I said, will contact many different amino acids. It's really a surface interaction at the end of the day, which increases by orders of magnitude the specificity of the binding. What the problem is that by increasing the specificity, you lose the intraspecies relationship that you have with small molecules. A small molecule binds to a kinase in humans, very likely will bind to kinase in virtually every mammal, and very often even in other species, faraway species like insects, Drosophila, and so on and so forth. An antibody that binds specifically to a human epitope will likely not recognize the same protein in other species. Very often not in rodents, just because very small differences will be enough to decrease the affinity by many folds. But right now, we are getting to the point that even non-human primates are unable to really recapitulate what happens in humans because the antibodies will not recognize the non-human primate protein. So this leaves you with in vivo systems where the target is not recognized. So the drug is essentially non-active in those models. And what do you do then? I mean, you can't use these for most of your experiments. Another fascinating example, and this is not really on the binding of the antibody to its target, but rather on the biology that regulates antibody stability. There is an active process of recycling of antibodies in all mammals that goes through a receptor, which is called FcRn, which binds to antibodies in lysosomes and recycles them in the serum. Now, this interaction is not perfectly recapitulated in any preclinical species, a little bit in non-human primates, but definitely not in rodents. So all the PK experiments that were used to run in different preclinical species now cannot be run in those directly. And this is a big conundrum for drug developers up to now. And that's why you need now to humanize specific aspects of these models.
Yes, so following on from that, and perhaps the harder question is, how is that resolved?
There are many different ways. Obviously, there's been an expansion of the use of in vitro model or ex vivo model. You know for sure organoids are becoming more and more important, but no matter what you do with these in vitro models, as complex as they can be, they will never recapitulate what really happens in an organism. So what we are doing is now we are exploiting the fact that we can tailor the mouse genome to really introduce the characteristics we want in the model. And as I was mentioning, we can either humanize the mouse genome by introducing the human gene. This will solve the problem of the presence of a target sequence, but we can go further. And what has been done at Jackson and other places is we can create highly immunodeficient mice that can be engrafted with human cells so that the human immune system will repopulate the mouse. Now, these models have the targets that are against which the antibodies or biologics have been raised, and therefore now you can again test them. So we remake these models relevant again for preclinical development. So definitely we're moving away from using wild type models, and we are moving more and more towards highly engineered models.
Excellent. Thank you for that answer. What does the current shortage of non-human primates mean for drug developers, generating the preclinical pharmacokinetic safety and efficacy data required to file investigational new drug applications?
Well, the short answer is that it's making drug developers' life very, very difficult. And the reason is because non-human primates have a lot of resemblance to the humans in terms both of physiology, but also of sequence of their targets for the antibodies. That's why they're the species of reference for toxicology, safety, more than efficacy, and we can get there if you want. So the idea is that you have an antibody that activates the immune system. And that's another interesting discussion that we might have later on why antibodies are so tricky to work with, because they activate the immune system. So you know what the primary target is, but you have no idea what's going on afterwards, because the immune system is such a closely related network of functional cells. But when you activate one part, you really don't know what's the network effect will be, what's the waves that will actually generate within the immune system. So let's say you have this antibody, and let's say that you're lucky enough that it interacts also with the same target in monkeys. But the idea is that relatively simple, right? You treat the monkeys, you can identify, for example, the maximum tolerated dose, and you think that, okay, that's what happens in a monkey, and you expect a similar dose to become toxic in patients. This gives you a reference point. You know you should not get close to that dose when you inject humans, at least initially. But now, if you don't have the non-human primates, you are missing this reference point. You don't know. You really have no idea, because you can do your in vitro works. You know which avidity is your antibody, what's the affinity of your antibody, you know how much antibody you need to saturate a certain percentage of your target. These are all required information, obviously, but it doesn't tell you what's going to happen to this complex network in vivo when you actually engage your target. You have no idea. And now just put yourself in the shoes of a drug developer that is going to inject for the first time a molecule that activates the immune system in a human being. Without knowing what happened. And I mean, we all know what happened with the Tigenero 1412, which is a CD28 activator that was safe was even safe in monkeys, by the way. It was even safe in human primates. And well, 6 people in emergency care with the very first dose, which was very low. So you can imagine you're injecting a drug in a human being for the first time, and you have no idea of the effect. So what you do is you actually start at very, 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 very low doses. You have these very conservative dose escalation, first in human clinical trials. There is this so-called MABEL approach. Essentially, what you do is you get all the in vitro data that you have, and you try to think, okay, what will be the minimal biologically active dose that gives me any kind of signal in the patient. And I start below that, and then I start escalating. Now, from the practical point of view, this means that this dose escalation means that you generally start with orders of magnitude below the efficacious dose, not the toxic dose, the efficacious dose. So before you actually see an effect, your phase 1 study can last for 2 years. But this is a big investment from your side, but also it delays a drug that might be working. And so it has a lot of implications from the clinical side as well. So all of this to say that it has a lot of complex repercussions, but mostly it's both the drug developers and the patients that are suffering because of this issue.
Yes, thank you. So how do you assess the efficacy and safety outcomes then?
How do you assess? I'd like to break this question into efficacy and safety. Obviously, that's kind of and actually, I would like to go back on the discussion we had on non-human primates, because non-human primates are relatively good if you have a cross-reactive, an interspecies cross-reactive reagent, are relatively good for safety. but they're definitely not good for efficacy. Because efficacy, and we are talking mostly about drugs in immuno-oncology here. I don't want to expand. I mean, it could be, but I really want to focus on an application that where these reagents are, I would say, the most advanced. Efficacy is relatively straightforward initially, right? You implant a tumor and you let it grow, and then you treat the animal with your drug, and you see if your drug is slowing down the tumor. So the very, very basic readout is a tumor growth inhibition index. But in reality, with these models, you can do much, much more, because you can see, for example, depending on the activity of your drug, you can see if you're modulating the tumor microenvironment. So it's not only the fact that it's inhibiting the tumor, but you can really look if it's inhibiting the tumor through the mechanism you're predicted, or if there is something else that goes on. Even if you don't have tumor growth inhibition, you might have some readouts that shows that your drug still works. For example, checkpoint inhibitors, as you know, are, well, right now they're the blockbuster, right? Keytruda, I think, hit $20 billion, $25 billion sales last year, and still by itself is only active in about a quarter of the patients. But in reality, the molecular activity is always there. So it always blocks PD-1, the PD-1 axis. It's just that it doesn't, that's not enough to inhibit the growth of the tumor. That's why all these combinations are coming out. So you can test that, right? Maybe your drug is not enough by itself to block the tumor, but is still acting on a certain cell population. And you know that now combining with something else, you will see activity. So it really depends on to test the efficacy of your drug, you really need to think ahead. What is the real readout that you want? What is the most important readout for you? Is it tumor growth inhibition? Is it target engagement? Is it cell engagement? Is it modification in the tumor microenvironment, and so on and so forth? And these are things that maybe with monkeys already, non-human primates, it's difficult to make because you don't have access to many of them, as you know. And actually, sorry, with monkeys, you can't do, now that I think about this, because you can't implant a tumor in a monkey. That's it. You don't have. The monkeys are not, for obvious reasons, and I think it's actually a good thing we can do that. For safety, things are getting much more complicated, because you have multiple levels of potential toxicities, right? You could have an acute toxicity, bispecific antibodies, for example, that engage T cells will very often lead to cytokine release syndrome, due to the fact that you hyperactivate these immune cells to release a lot of pro-inflammatory cytokines, and you have this network effect we discussed before. We have these ripples, these waves that really go through the immune system and will activate multiple cells that you may not want to activate. So it's an acute effect. You inject these reagents, and the patient or the model, hopefully before, reacts immediately. But you have also long-term effects, like cytopenia. You're killing certain cells in the immune system, maybe, and you need to really think about readouts to follow this longer-term effect. You might know that now CAR T cells have been black boxed because there are some questions about potential secondary tumors. Now, we know this is very rare, but it's a possibility, especially for CAR Ts that have been highly engineered. So safety, in this case, humanized mice can help you addressing these questions, for example, by following for 6 months, a year, these humanized animals with your CAR T. So I'm not sure I answer your question, but because it's complex, but the question was complex, therefore the answer was maybe a little bit long.
No, that's absolutely fine. You definitely decrypted what was a very complex question, so thank you for that. So I think we've touched on this in part, but my next question is, what impact will humanized mice have on future tools for preclinical development of drugs recognizing human-specific targets?
That's only what the future will tell, right? I mean, we can only speculate. But there is definitely a trajectory. We know humanized mice have been adopted by drug developers in a very important way, I would say only in the past 3 to 5 years, not before. And there are two factors that are coming up here. First of all, more and more drugs are in the antibody format and the biologics format. So they have the issue that I was mentioning before, which is you do not have a good cross-species reactivity, and you need therefore human target in functional human cells. So this definitely will drive more and more, I would say, the use of these animal models in preclinical research. The other aspect of this, the flip coin, is that when people think about humanized mice, especially people of my age, I would say, which have been in the field for quite some time, they think about the early stages of humanized mice, where these humanized mice had either a very defective human immune system, where only maybe the lymphoid compartment was partially working and there were no myeloid cells, or where human cells would mount a reaction against the mouse tissue, provoking a general activation of the immune system that would kill the mouse. So those times are gone. Those were the first models. We're now at the fourth or the fifth generation of these models that are much more complex and much more useful. And I think this usefulness will really increase in the predictability of the results you get with these models. So if I can see the future of these animals, or if I can imagine the future of these models in preclinical research, there will be more clinical-like trials in these models. For example, multiple donors will be used in a single experiment to mimic what the human population, how the human population respond to the drug. And I really hope that there's going to be also a feedback from the clinic to the preclinical field, asking them to really design experiment in ways that are more similar to what the clinics look like, because I am strongly convinced that these models can be really, really predictive. And I'd like to go off on a tangent here a little bit, if I can.
Of course.
Which is the traditional issue of drug development. And this was something that was an issue both for clinical developers, but also investors, for example, and other actors in the drug development field, was that most of the drugs would fail at the clinical level, and therefore it was very difficult to understand which drug to focus on very early on. But what I'm seeing now is that now that we understand much better the mechanism of cancer and how to address them with very, very highly specific reagents, actually the clinical success is getting a little bit more consistent. If you think about phase ones, for example, a classic phase one for a small molecule is just a pure safety. Is this molecule going to kill a patient or not, or make them worse, or make them sicker? Right now, a phase one for an antibody that targets a specific tumor, investors, for example, are expecting to see efficacy already. And these are last line treatments, right? These are usually people that have gone through multiple lines of treatments, and this is just an experimental drug that they're trying. But there is already an expectations that these drugs work. So I think we have quite a few drugs that are working, that are hitting now, or they will be hitting the market soon. And now the question will be, how do you position your drug against the other drugs? How do you identify the patient populations where your drug will fare better or will be better than your competitor drugs? And I think animal models here, the humanized models, will be crucial for this, because you can then run multiple studies with different biomarkers and so on and so forth to predict this specific response. And therefore, I really think that these models will also help the clinical development of the drugs, not just the preclinical part.
Absolutely. Thank you. And now, while you've got your crystal ball out, I wonder if you could share share some of the key breakthroughs of recent years in the field and how these will impact future success and how they're currently impacting present success.
It's a tricky question. So I think the field has been growing more organically rather than by huge breakthrough. There are some general themes that we could consider breakthrough. For example, the expression of human cytokines in an immunoefficient background to allow for the differentiation of specific cell populations in a physiological way or paraphysiological way, it really helped making these models very, very relevant for drug developers. For example, the expression of IL-15 in humanized models allow for the differentiation of NK cells, of functional NK cells. So if you're working on an NK cell engager or on NK cell based therapies, this is definitely been a breakthrough for you, right? So I think that the other big line that is starting to make an effect is that finally drugs that have been tested in humanized mice are going to the clinic, and the proof is in the pudding. I mean, we can say as much as we want on the fact that these animals are predictive and so on and so forth, but now we start to see finally articles coming out where groups have done the preclinical work, saw some things, and then went to the clinic and saw the same things. So I think these are the breakthroughs that really will be driving the adoption of these models for the preclinical development, and are driving those in the next, I would say, at least 2 or 3 years. Then who knows?
Yes, then we'll have to compare notes in a few years and see if
Yes.
you're right. We'll have this evidence here. And so the last question that I have for you is, what do you think are some of the key challenges in the clinic that we need to overcome to ensure that the success happens in the next couple of years?
Well, I go back on what I was saying before on identify the right drug for the right set of patients. And the additional combination is identify the right drug combination. It's being clear now with the beautiful work that has been done recently on the cycle of tumorigenesis and in an immune system environment, that you can't really hit a single mechanism of tumor formation or immune system activity. You need to act on different fronts. You need to, for example, hit the tumor with an ADC, but then activate the immune system with checkpoint inhibitors, or maybe even engage T cells at the same time, so that you really are making sure that the tumor will not escape your therapy and that you are efficacious as much as possible. But with these, you will also increase the toxicities that you will see. And therefore, combination therapies are definitely the future of immune oncology. Also, with small molecules that are already currently used in combination with the new therapeutics that are coming out. But the big challenge will be to understand how and when to use a specific combination with which patients.
Great, thank you very much. And so that brings me to the end of my questions. I wonder, we've covered such a broad scope and you've given so much incredibly invaluable insight. I wonder if there's anything that we haven't mentioned that you wanted to discuss or...
Oh, okay. So that's the open-ended question.
Yes, that's the open-ended question.
Well, I think one of the things we have not really discussed, it's an underlying theme, but we have not really discussed it openly, is how this model, in my opinion, will change drug development or the process of drug development. Because right now, there is a relatively clear distinction between the preclinical phase and the clinical phase. So the preclinical scientist, they are making sure that the drug that they are developing fits within certain boundaries that are made by the regulatory agencies, by the FDA or the EMA and so on. So there are certain requirements that need to be followed. Fair enough. Then you get to an IND, and once you get the IND, the drugs become something else, right? It drops off the preclinical development. I think what we are going to see is more and more drugs that are already in clinical development going back to preclinical for additional modifications. And this is because, opposite to small molecules, an antibody has almost infinite variants that you can tinker with, right? You can increase the half-life, you can change the affinity to different Fc gamma receptors, therefore modulating its ability to interact with different cells in the immune system. You can modulate its affinity. In certain cases, as it's been seen, that lower affinities are better than higher affinity for engaging targets in specific ways. So I think that this model will really help blurring the boundaries between preclinical and clinical development and making it really a continuous drug development flow where you get lessons from the clinic and now you're going back to these models and use them to improve the molecules that you're already developing in humans.
Yes, thank you. And I think that's a great note to end on. Thank you so much for your time today and for your insight. I'm sure everyone will have learned a lot from that. I'm sure there'll be loads of extra questions. So we'll follow up with those if the audience do ask them. And thank you so much.
Thank you very much for having me. It was really fun.
That brings us to the end of this DDW vodcast episode, sponsored by the Jackson Laboratory. If you are enjoying this content, be sure to join Free Today to become a member of Drug Discovery World. Membership includes full access to the website, including free news and exclusive content, unlimited app access, a weekly e-newsletter, podcasts, vodcasts, white papers, ebooks, and information from trusted third parties. Thanks for watching.